It's my great pleasure to welcome Professor James Bendle today. James holds a master's degree in quaternary science from the University of London, a PhD from the University of Durham. I believe he had a few postdocs globe-trotting, including Japan, Bristol, and Scotland, before he joined the faculty at the University of Birmingham, where he's been since 2012 and currently is the equivalent in our system of an assistant or associate professor. I know James because I spent a few months in Birmingham in the latter part of 2019. He was my host. I had such a wonderful time there. He and I have a little project on a back burner since the pandemic that we're hoping to continue with. But I'm super excited to hear about some of the proxy work he's been doing, which I think is extremely relevant to our seminar today. So join me in welcoming James Bendle.
Thank you. Thank you, Brad, for that introduction. Thank you to Maggie as well for kindly hosting me and for a really interesting chat before, a fellow organic geochemist, and thank you for so I think it's Kate Freeman here as well, and I think this series is in honor of Kate, so I'm honored to be included as part of this organic geochemistry themed seminar series for Kate Freeman, so thank you. So yes, today I'm going to talk about work that firstly, can I just all check that you can hear me okay, and that you can see a normal slide PowerPoint view just before I go on?
It's all good here.
Okay, that's great. So thank you. Yes, I'm going to present work that we've been developing over about the last 5 years now on a whole new suite of biomarkers based on 3-hydroxy fatty acids and their potential application for terrestrial and marine paleoclimate. And this is the product of a number of different collaborators, which are shown here. So, okay, so what are 3-hydroxy fatty acids? So these compounds are part of the lipopolysaccharide or LPS coating that occurs in all gram-negative bacteria. These compounds are, although they're fairly new in terms of their interest for paleoclimate and geology, but they've been known to biomedical science for many decades because gram-negative bacteria are so abundant, and these are a common biomarker for those bacteria. And it's what they will look for 3-hydroxys in a medical context. For example, if you get very sick with gastroenteritis problems and you don't know why, and they suspect that you might have got contracted or come into contact with salmonella or another bacteria that acts as an endotoxin, they'll look for these 3-hydroxys. So they're well known to medical science, but ignored by paleoclimate until fairly recently. They're quite small compounds, so they're shown here on the left-hand side is the molecular structure, and they tend to have 10 to 18 or so carbons. And so if you don't understand that the chemical shorthand, these structures simply show the carbon and hydrogen backbone. On the right-hand side is the main functional group. So this is the 3-hydroxy acid group. But what I want to draw your attention to is the fact that they come with in a number of different structural variations or isomers. So there's the normal compound here, which has this straight chain of carbons and hydrogens. Here is the isoisomer, and here is the anti-isoisomer. And these just have an additional side branch here and here. But what that does is it changes the melting point and the fluidity of those compounds. And so that's where they come into interest for us, because the bacteria likely change the composition of these compounds to regulate membrane fluidity in response to the temperature of the environment and the pH of the environment that they're living in. So we now know that they occur widely in the environment, in soils, lakes, marine settings, and that they are preserved in geological archives, in lake sediments, in stalagmites, and in marine settings. And this is something that we are really actively exploring at the moment. And we're very interested to discover how far back in geological time they're preserved and you know how far back they can be used reliably. But I think at this stage, I'm very confident that they will have applications for the late Quaternary at least. So I think it's useful to go back to the earliest studies and explain why we settled on these compounds, why we decided to explore them. And it's also probably the easiest way to introduce their chemical underpinnings for how they function and how we can use them as geochemical tools. So it actually derived out of when I first moved to Birmingham, the head of school was someone called Ian Fairchild. Professor Ian Fairchild was a big person in the stalagmite community. And although I'd worked on biomarkers in marine settings, in lakes, and recently published in ice cores, I'd never done stalagmite work, and I didn't particularly want to be honest. I felt overstretched as it was, but he was very keen that there was biomarkers were there in some stalagmites, and they hadn't really been exploited properly. And so I did agree to a collaboration with a group at CUG, Wuhan, China. And took on a PhD student, Chanfa Wang, and we were finding these three hydroxys in real abundance in the stalagmite samples, but didn't really know what to do with them. So in stalagmites, there had been some publications on GDGTs, Tex86, and they hadn't really worked very well. There've been some studies that showed that plant waxes were preserved. And in this HS4 stalagmite, which is shown here, there's this guy standing next to it. It's this huge stalagmite, and it's quite organic rich. There was really good plant wax signal, and that's what we initially wanted to do. And we did publish an isotopic record of plant waxes. It's the first time that anyone had published compound-specific isotopes on plant waxes from a stalagmite. So that was how the project started. But then we kept finding these three hydroxys, and then thought, okay, do they have some temperature signal encoded? Because when we started to look at the older literature, you could see that they did have, the functionality of them was controlled by or responded. Sorry, the functionality of them meant that they had very different melting points, and so we took a set of samples from a mountain transect in China, Shennonjia Mountain, which is in this Hubei province of a very, very steep topographic area in China. And if you've ever seen those YouTube videos where they have, these scary glass walkways above these, terrible, like vertigo-inducing drops, that's that area of China. It's not actually the exact mountain where we took the samples from, but it's in this region. And it's kind of a very simple way of testing whether there was, an environmental signal or a temperature signal encoded by these compounds. So the theoretical underpinning why we thought there might be potential is because the anti-iso compound, for example, so this shows the compounds with 15 carbons, and the anti-iso isomer has a very different melting point compared to the normal isomer. So the anti-iso fatty acids have a lower melting point, and thus we thought that the bacteria living in colder temperatures may be producing more of the anti-iso compounds compared to the normal compounds. And that was something that, looking at the stalagmite samples, although it's very subtle, we thought maybe we could see something. But then we got the results back from this mountain transect, 26 soil samples, and very clearly we could see this. What I should say here is that we came up with an index, to a very simple index that just expresses the ratio of the anti-iso to normal compounds with 15 carbons. So that's called RAN15, which stands for ratio of anti-iso to normal fatty acids with 15 carbons. And the higher the index, the more of the anti-iso compound. And so what we could see is as we went up the mountain, so following the temperature as you go up the mountain, decreases from 14 degrees to 2 degrees centigrade at the top because of the, it's controlled by the local lapse rate. It's a mountain that's several thousand meters high, so by the time you get to the top, it's much colder. And lo and behold, the bacteria or the soil samples that we were collecting from the top had much more of the anti-iso compound in, as expressed in this RAN index. So going from nearly 2 at the bottom to 8 at the top, which means there's much more of the anti-iso compound. We also looked at we also just searched through all the other ratios and tried to search systematically for other ways that these compounds may be changing along the soil transect. And the other thing that we're measuring as well as temperature is pH and soil moisture. There was no correlation to soil moisture. pH in this mountain transect had no correlation with temperature. So pH was completely independent because I think it's quite a complex local geology. So that was very useful because those two environmental parameters were independent. And then what we found was that there was another correlation to soil pH. So this is the RINE index, the ratio of iso and anti-iso to normal compounds. So for temperature, we found that it's on that mountain in China, only these two compounds, the anti-iso and normal 15s, had the systematic response to temperature, but all of the compounds, the full suite of compounds, were varying systematically in response to pH. So there was more mountain transects were published after we did that initial paper. There's a group in France that's also been working on 3-hydroxys, and again they found that a similar change in mountain transects with these compounds, so more of the anti-isos at colder temperatures. But they did several different mountains and found that there was, although there's this trend is seen, that there is a difference in slope and in the intercept to some extent, suggesting that in these different regional settings, maybe there's a different bacterial biogeography, and that is also playing a role. So that you can't necessarily, so at that early stage, it suggested that it's important to undertake a local calibration study or to understand the local context. Anyway, having done that for this mountain in China, we applied our new calibration to this HS4 stalagmite, and we also, at the same time, were looking at the plant waxes, which was the original motivation for the study, and their isotopic composition. So just to explain why we were doing that, C3 and C4 plants are the two main plant types, and they produce plant matter, plant material with a different isotopic signature. C4 plants are more isotopically heavy, and C3 and C4 plants respond to changes in climate. And China, Southeast Asia has a mixed assemblage of C3 and C4 plants, so which should be quite sensitive to changes in climate. So this is the downcore reconstruction from Heshan Cave for the Holocene. So the present day is on the left-hand side, 9,000 years ago is on the right-hand side. And the very top of this reconstruction in this thicker purple line, this is the oxygen isotope curve from that stalagmite. So this is the classical approach to reconstructing climate from a stalagmite, and oxygen isotopes measured on the carbonate of that stalagmite. That encodes, that captures both a signal of both temperature and rainfall. And it can be quite hard to separate those two signals out, which is one of the motivations for looking at biomarkers, maybe that record temperature or pH, which may be related to hydrology separately. Below that is the change in insolation in the Northern Hemisphere. And this record, this isotope record, has been interpreted as there being a stronger monsoon and warmer conditions in central China in the early Holocene, and then a shift towards cooler conditions. And then you can also see some oscillations across the medieval warm period and the Little Ice Age. The record below in purple here is lower resolution, because it is harder to make these biomarker measurements compared to an isotopic, an isotopic measurement on the carbonate. So the resolution is lower, but this was the first independent temperature record based on biomarkers from a stalagmite. And it gave really quite reasonable results for such a new technique. We found that the temperatures reconstructed, which is shown here on the left-hand side, the most recent temperatures are on the order of 18, 19 degrees centigrade. Mean annual temperature measured at the cave using data loggers is 18 degrees centigrade. So the absolute temperatures reconstructed by this technique were really quite reasonable straight off the bat. It reconstructs a warming in the earlier part of the Holocene of fairly subtle warming of about 1 degree centigrade. So, you'd expect in the Holocene fairly subtle changes in temperature. We see a longer term monotonic cooling, and then we see oscillations, in temperature, warmer medieval warm period and a colder little ice age. So the temperature reconstruction showed a lot of promise for such a new technique. We also reconstructed qualitatively changes in hydrology based on the pH proxy and inferred wetter earlier and wetter later Holocene with a drier mid-Holocene. And this is something that is actually seen in many records in central China. It's kind of unique to central China that there's a dry or a failure of the monsoon or a drying in the mid-Holocene. Then we also, based on the biomarkers from that stalagmite, reconstructed a change in C3, C4 vegetation. So qualitatively, more C3 vegetation in the warmer, wetter early Holocene, drying through the Holocene, and more C4 vegetation due to the heavier carbon isotopes of the plant waxes that we recovered, which is sensible. We also inferred changes in bacterial substrate selectivity. We're interested to see if we could get constraints on how bacteria are responding through looking at the lower molecular weight normal fatty acids. That's quite a complicated story, how we interpret this and how we arrived at that, but that was published in EPSL. So I won't go into that any further, but I'll just put that out there if that's something you want to read up on. So that's how we started our journey with these new compounds, and then subsequently got funding and a number of PhD students working on looking at these compounds globally and continuing to work with the Chinese as well. So we compiled from repositories a global compilation of soils, so shown here in the top left, that cover a much bigger range of environments and temperatures, and that's something that we published on. We published a global soil calibration, and I'll look at that next. Something that is ongoing is we did several road trips in the US. We've also been working in Tenerife, and then even more recently Southeast Asia, to collect fresh samples from lakes, from soils, and collecting these fresh samples is important rather than the archived soil samples, because we're able to freeze them in the field, to take lysis tubes to preserve the DNA, so that when we publish this work, we'll be publishing both the biomarkers and for every sample a bacterial ecology as well. So essentially, we'll have the bacterial biogeography as well as the biomarkers for these transects. That's work that's ongoing. So this is from the global soil calibration that we published in GCA, and here you can see shaded the temperatures from the soils that we collected, that we analyzed. And if you look at these boxes, these inset boxes, what you can pick out is how the anti-ISO 15 increases systematically from warmer through mid-latitude to colder soil. So much more of this anti-ISO here in the soil from Greenland, for example. So just showing in a kind of visual way that response. This is the regression that we obtained for this round 15. So this is the increase in the anti-ISO isomer at colder temperatures globally for 185 samples. There's the residuals below. So there's a fair amount of scatter in this relationship. So there is a global relationship, but within that there is regional scatter, and there's a lot of scatter at the cold end of the temperature spectrum. It sort of breaks down below about 5 degrees, which is something that we've actually is a common challenge for biomarker temperature proxies. This is the RAN 17. So the other compounds that show a systematic response to temperature in soils are the 17s, the homologs with 17 carbons. And that was a similar story, really. A bit more scatter in the mid latitudes, possibly behaving a little bit better at the colder end of the spectrum and at the warmer end. But then when you look at this regionally, so this is showing the regional correlations. You can see that there is, some real differences in the intercepts and some differences in slopes as well. And this is emphasizing that really, I think if you want to apply these compounds to a specific lake or cave context in the future, a local understanding, local constraints, and a local or regional calibration is going to be really important. If we try to apply this global soil calibration to our HS4 Chinese stalagmite record, we would have got temperatures that were, way too warm, way off. So in that case, we had to use the local calibration was really important. This is the pH proxy. So this actually shows a tighter relationship globally, but a very clear relationship there across a big pH range. In the global soil calibration, we also applied machine learning approaches. So this is something that we've been working on for a few years with our collaborator, Ilya Mandel, who's an astrophysicist and mathematician who uses machine learning to look for to sort of try and extract the signal of from distant nebulas and filter that out from all the noise, all the background noise. And he sort of worked with us as a side project very kindly, and was surprisingly enthusiastic about it. But we first worked with him on machine learning approaches to improve the calibration for the different biomarker approach called TEX86, and then we applied the same techniques to these 3 hydroxys. So what the machine learning does is it looks at all the different compounds, and essentially it's looking at how the different compounds behave in response to as many different environmental parameters as you can input, and it's sort of holding all that information together and comparing very subtle changes. And then it produces coded calibration. So it's not a kind of simple calibration that you can it doesn't collapse things down to 1 or 2 compounds. It's throwing everything in there. And what comes out of a black box is a somewhat better calibration. So using the machine learning approach, it gives a better R-squared value, a pseudo R-squared value, and a lower error for both temperature and pH. So that shows, something that we should continue to explore. And also we work with Sarah Green on this to host this in a way that's very accessible. So all the code for both these 3 hydroxy calibration, but more importantly, the optimal one for TEX86 for the GDGTs, which is a much more established technique that many people want to work on. That's all hosted on GitHub and is very accessible. You can easily put in your own results and get the machine learning calibration results back. So how am I doing for time? I'm good. I'm at 26 minutes in. Okay. Okay. So the next logical thing after doing this work in soils and stalagmites was to consider, how these compounds preserved and what potential do they have in marine and lake settings. So the next thing that we did with another Chinese PhD student. So by this stage, I was hosting 2 PhD students, are splitting their time between CUG Wuhan and Birmingham. And Yang Yi came to work with us, and then he also went on to work with Anne Pearson and Felix Elling as well afterwards in Harvard. But he produced the first marine calibration, so working on a series of core top samples from the tropics in the West Pacific along these coastal basins to the high Arctic. So quite an impressive temperature range from 1 degrees to 28 degrees centigrade. And he found in the marine sediments that the 3 hydroxys were there. They're well preserved. The overall distribution, the overall fingerprint is somewhat different from soils. There tends to be more of the shorter chain 3 hydroxys and more of the anti-iso 3 hydroxys in marine settings. At this point, we started to do some DNA work on a limited number of samples. So the current work that we're doing for the freshly collected material from the US and Tenerife and other places is going to be like every sample is going to have a corresponding bacterial ecology with it. But in this case, we just had a few representative samples. But you could already see that there was a difference between soils here on the left and marine setting. And the marine setting has a simpler bacterial community dominated by proteobacteria with some Verrucomicrobia, and the soils is much more diverse, which makes sense considering how soils are heterogeneous compared to marine setting, I guess. Now, maybe because of the more simple ecological setting in the ocean, I'm not sure, but the marine calibration that we produce straight off the bat has a very impressive R² value. It's an exponential function here. So here we're going from 30 degrees centigrade to below freezing, and this is based on the compounds, the homologs with 13 carbons. So we tried the soil. We tried to apply the calibration to the 15s and the 17s that we were finding in soils that have this systematic relationship to temperature. In the marine setting, it didn't work. So whatever is producing 15 and 17 in marine setting, it's not recording temperature in the same way. But we did find that the C13 compounds change systematically with temperature, and this is the relationship, which actually, compared to the main sort of biomarker-based marine calibrations, UK37 derived from alkanones and TEX86, this is actually really impressive. There's a bit of a data gap here in these lower, cooler to medium, mid-latitude temperatures. So I'd like to see what's going on here. But certainly at the cold end of the spectrum and at the warm end of the spectrum, this is quite well behaved, and I'm quite excited about the potential. Of course, it needs to be applied to sediment cores to really understand, and prove that potential, and that's something that we want to do very soon. The other thing that we then did is start to work in lakes. So this is the bigger kind of campaign at the moment that's ongoing. So we already published with the Chinese PhD student Yang Yi the first lake calibration. This is all alkaline, relatively alkaline lakes in China. And what he found was that the distribution, the fingerprint of these compounds in lakes sits somewhere between what we'd seen previously in soils and in the marine settings. The RAN 15 and RAN 17, so the two approaches that had worked in soils showed a relationship to temperature we used in the stalagmite, did not correlate with mean annual temperature in these lake environments. The RAN 13 did have a reasonable correlation. So this is the same compounds that we find in marine settings. And then he also found that the ratio of iso to normal 17 compounds had a relationship to temperature. There's also a small amount of DNA work done on these in these settings as well. So we can now compare on the left here soils, in the middle 3 samples lakes, and on the right hand side marine samples. So this is showing the bacterial distributions at the phylum level. You can clearly see differences, and then at the class level, which highlights, superficially you could say that the marine ecology is much lower diversity, and soils and lakes look relatively similar. But when you look at the class level, you can see, that's emphasized even more, and there is clear differences between soils and lakes. So, for example, in lakes, these bacterioidea are much more abundant, uniquely abundant. So all of this is suggesting that, in going forward for the application of these compounds, that local regional calibrations are going to be necessary. And if you want to reconstruct temperatures from a lake sediment core or marine sediment core, you're going to need a lake or marine calibration, not going to be able to use the soil calibration. So these are the transects in the US that we're working on. A temperature transect, which is East Coast soils, going from Maine down to southern Florida. And then we also did a road trip, which I was lucky enough to take my whole family on to help take samples all the way across from Louisiana through to the hyper-arid areas in the Southwest. And this is much more of a hydrological gradient, and extreme isotopic gradient as well. And that both these transects are following published transects, so they're data rich. So plant waxes, GDGTs, etc., have been studied along these transects, so we can then directly compare what comes out of our 3-hydroxy work with that published data. And that's something that is just, it's very, very close to being. It wasn't quite ready to share it at this meeting, but hopefully by the AGU, the students will be presenting some of this data. We've also been working on Tenerife, which is one of the Canary Islands. The advantage being that, compared to getting to the US or other places, it's relatively close to us and relatively cheap as a popular holiday destination for people from the UK. And it's also a very, it's one of the biggest mountains on the planet, right? And it's very steep, goes through a number of different ecological zones. So in a very, very small space, you can get very extreme changes in climate and ecology. This is just showing some of the sites that we were sampling in the US, taking both lake samples using ecmograbs and also soils from the surrounding catchment area. We went, and this is Tenerife. The work in Tenerife, we've been putting down these probes, these data loggers, which I recovered last year. So we also have in situ soil temperatures. So we're not just, in the case of Tenerife, we don't just have to rely on weather stations and interpolating between weather station data. We've actually got in situ soil measurements over several years. And this is Alice and Amy, her 2 PhD students, who've been doing parallel projects with Amy working on the DNA and Alice working on the biomarker elements. Another nice thing about working on Tenerife is that it's this is showing the changes in the vegetation zones, biomes that you get on either side of the island. But it's been recognized for a long time that it's a great place for biogeochemical studies for these reasons. And actually there's a nice kind of historical context there that I think Kate and Maggie and other organic geochemists would appreciate, and that this is where Jeff Eglinton went in the early 1960s, took a sabbatical when he was at Glasgow, and collected leaf waxes from these different biomes, hoping at that time that actually the leaf waxes could provide a taxonomic fingerprint of different plant types, which actually didn't happen. We now know, of course, that plant waxes do maybe change in response to temperature and humidity, but they don't really have a taxonomic fingerprint. Oh, by the way, as well, I did talk to Jeff about this, and in the early 60s, when you got a NERC grant, he got a NERC grant that paid for this work and this sabbatical in Tenerife. It included a valet and a driver for the whole time that they were there, paid for on the grant. Times have changed. Okay, so just a few more results from our ongoing work. So this is bacterial community structure from several of the lakes in the US. So at the sort of 2 ends of the gradients that we're working in the Southwest. So Mary's Lake is in New Mexico, but actually up by the border with Colorado, very high altitude alpine environment. Lake Seguro is near Phoenix in Arizona. And despite those very different environments, the lakes have quite a similar bacterial community structure, which is promising if you think of, we're going to be able to develop calibrations, lake-based calibrations, and apply them broadly to different lakes. That's promising in that context. And there's very clear differences between the surrounding soil bacterial community and the bacterial communities in the lake, with some differences between the two lakes as well. So this is just showing some of the GCMS results, some of the traces, the different geochemical fingerprints for those lakes and soils. And then Amy's been applying this amplicon sequence variant technique for differentiating between bacterial communities, and essentially this shows that there's a complete separation between the bacterial communities in the lake sediments and the soils for Lake Seguro, and a very little shared community structure in Mary's Lake. So really emphasizing that lake-specific, calibrations are going to be critical for developing this proxy for work in lakes, and that's something that we're doing at the moment. Then the final thing I wanted to highlight was that we're working on bacterial cultures. So Amy has been isolating bacteria from both lake and soil samples, because we want to know which species produce specifically which 3-hydroxy homologs, and can we do lab-based culture experiments? Can we vary temperature systematically and get lab, culture-based temperature calibrations under controlled conditions? So she's doing the controlled temperature experiments at the moment, but she has successfully, isolated, extracted 3-hydroxys from these pure bacterial cultures. And what that's showing us is that the fingerprints that we get from soil and lake settings is, as we suspected, an aggregate of inputs of 3-hydroxys from different bacteria. It's not that all the gram-negative bacteria produce broadly the same 3-hydroxy fingerprint. That no, certain species, certain classes are going to be producing more dominantly certain 3-hydroxy compounds. So it may be that those, when we're using the 15 compounds that have this temperature relationship in soils, that is really being produced by a limited number of the bacteria. And same thing in soils and marine settings. And if we want to produce more accurate reconstructions using these compounds, then we want to try and isolate those bacteria and understand what is producing the 3-hydroxys in those different contexts and settings. So in summary, we have independent temperature and pH proxies. It's early stages, but we're able to demonstrate the viability, I think, for 3-hydroxys. We had the first successful applications to a stalagmite or any archive. We're currently exploring them in a number of different lake settings. We have the first marine calibration published, and global terrestrial soil calibration, the first regional lake calibration. So in progress, we have soil and lake transects pairing DNA with the organic geochemical work. So we'll be doing the bacterial biogeography and the biomarkers at a much higher level of detail than we've done previously. And we're also doing culturing work now and starting to really explore downcore preservation and applications in a number of different contexts. So thank you very much. And I'm happy to take any questions.